About 3 hours after creating this sealed wet mount slide, things look pretty much as they did. There are diatoms slowly moving about the slide, which are at the low power setting, the most apparently dominant of the organisms to be found on this slide. Although the biodiversity, as usual with this lake, is very, very high. Nothing is really much happening. The sealing layer of petroleum jelly, which you can see here, I'm following along the side of, appears to be intact. I don't see any compromising with the seal, which is a common thing to have happen. A single break in the petroleum jelly gasket between the slide and the cover slip will cause evaporation over time, and depending on the severity of the break, it can be anywhere from minutes to days. I suppose days, hours at least. But this one appears to have gone off well, and here we go. I keep stumbling into you. I believe this is a rotifer, but I really don't know. The largest organism on the slide that I've found so far. It's a pretty good look at him. Chase him around a bit. Anyway, yeah, slide's doing very well. Let's step up the power a bit. Refocus. Okay. There you can get a good look at some diatoms. Much more detail now. Let's see. Just typical freshwater lake organisms, and nothing seems particularly unusual about the population distribution, of course. Nothing seems particularly unusual about the level of activity. It's very average at this point. Of course, it's also very early into this slide. Long, long way to go. Let's do you. Higher power. Focus. 40 power objective. And very carefully bring it back in. Okay, found a diatom. That, I'm hoping, is at the right level. There, I found her. Okay. It's mostly luck, believe me. Now you can get a good look at the internal structures of that diatom. And let's mess with the condenser focus a little more. Bring out the details better. Condenser focus makes a huge difference to what you see in the microscope. A microscope without a condenser focus just really will not give you the kind of level of detail and clarity that one with such an adjustment can yield. See this fringing around the edges? That's common to microscopes that are badly focused in that way, and there is no fix for that other than to correct the focus. See, it's kind of a ring effect. It's a diffraction taking place. The light rays are being refracted to the sides, and you can correct for that with condenser focus. See? Very neat, huh? You lose contrast, but that can be corrected in software. So there, very nice details, huh? It's beautiful. Very good. I'd like to see that under oil immersion, but I dare not do that because once you oil immerse a slide, a delicate slide, you're done playing around until you can be brave enough to try to clean the oil off of the cover slip surface, which is a very, very dicey operation. I've done it before on slides like this, but it's tough. Oh, it's tough. It's an ugly, ugly prospect. Let's just kind of drift around and see what else we can see. Look at that there, that's a bacilli. Off center there. Bacillus Bacterium. Very clean example of a Bacillus Bacterium. Usually there's a little bit of a warp to them, to their shape. But these are very clean rods. What species? Heck if I know. I am not that good. That's probably another bacterial chain. You can see some slight segmentation there. A little fooling with the condenser focus, and yeah. Now it's hard to see because this is a very small bacterium. This is a very small organism, and thus harder to pull the details out without jumping up in the power. But if you look real close, you can see it's comprised of about 5 segments. That's a bacterial chain of some kind. Could be blue-green algae bacteria, could be something else entirely. This here appears to be a large, fat type of Bacillus Bacterium. I've seen those before, something like them anyway. If I'm right, then there will be colonies spring forth off of that single microbe, if it's viable. This here is a Diatom, long, thin one. Starting to work with objects fine enough that they're testing the limits of this particular objective. They are no challenge, period, for the higher power oil immersion objective, but as I said, I'm not going there. Something on the slide there. Oh, hello. Long bacterial chain. These are common. Long chains of bacteria. See the dots? Those are organelles of some kind. You and I, we have organs, which are composed of billions of cells. Single-celled creatures have organelles, which are, the organ equivalent to a single-celled microorganism. They provide cell functions, processing of nutrients, asexual reproduction, that sort of thing, cellular division, or binary fission in the case of bacteria. And what you're seeing there, these periodic dots, my guess, because I can't really make out the segments here, is that there's one per cell. One per body. Chunks of bottom sediment usually thrive with at least bacteria and often attract large numbers of protozoans. Here is an actual living, very tiny protozoan. Take a close look. Watch that bugger. See the stuff moving inside? It's not simply rotating, although that's part of it. Those organelles inside that cell are actually moving around, doing their thing. They have to move from point to point within the cell body in order to maintain the vitals of that cell, the vital functions of the cell. Each little globule in there has its own specific purpose, much like the organs in your body. The organelles inside the cell are vital to its continued survival. Quite fascinating, isn't it? Oh, look at this here. Some bacteria. These are Bacilli bacteria, a pair of them, doing their thing. Bacteria often have flagella, and this is what they use to propel themselves from point to point within a fluid medium. They whip them, much like a whip, in a corkscrew fashion, and it propels them throughout the medium wherever they need to go, which is generally toward a source of food. Interesting that I found a pair of them. It would be a safe bet that this pair is actually bound. Their flagella are probably stuck together, and they are actually clone copies of each other. They're probably each other's offspring. One probably underwent binary fission and gave rise to the other. Which bore which? Neither. Both. They split in half. Their material splits in half. Their genetic information splits in half. Neither one is the father, neither one is the mother. They are both and neither. And a large chunk of crap. This is really what we came to see. Because within this should be a lot of motion. Organic debris attracts organisms that both serve to decompose it and serve to eat the things that are decomposing it. Well, that was fast, geez. Faster than I intended. And there it is. And you can see, yes, a lot of motion happening around the outside. Those larger protozoans here. Oh yeah, hide on me now. Those larger protozoans are attracted to much smaller organisms which they can consume for food. Like this little guy here. Boy, I would like to get him under the higher power. Wonder if I can do that very quickly. Got him. There you can see one of those fast-moving large protozoans. This guy is very green. That looks good, huh? You can see things move a little bit in there. Common to these larger organisms are structures called vacuoles. They're pockets within the cellular membrane, within the protoplasm that makes up the internal goo of the cell. And these can expand, contract, and move, and that's the way they transport nutrient throughout the cell. Here's a little guy. Little green guy. That could be Brownian motion making it move. No. Zoom, he's gone. Fast little guy, huh? Gone in a flash. Oh, look, this is interesting. This diatom appears to be growing things. Those may be some kind of bacteria that are feeding on the diatom. They're very clearly semicircular shapes. like a letter C, and there's several of them. There they are, more of them. So these are discrete organisms. See that? Definitely discrete organisms, some type of large-sized bacterium. Really unusual. Oh, you're interesting. Strange looking organism there. It's oddly shaped. Look at that. It's like a swimming Dorito. That's really weird. And another first. Never seen anything like that. Okay, here we go. I call this superworm. This is actually not a worm at all. We humans are conditioned to look at anything that's long and thread-like and very obviously moving as a worm. But this, if you look very closely, you'll notice it looks like it's got scales. Well, that's not scales. Those are segments. And that's why I need to get on this one really quickly before it wanders away, because I need to see that at a little bit higher mag. Okay, a lot higher mag. I think I got it. Okay, yeah. Boy howdy. There's your worm. And this dude, as I said, is not a worm at all. It is actually a chain of single-celled bacteria. At some point, at some place that you cannot see because these details are too weak, there are flagella on this thing that it is whipping, apparently in unison, in order to move through the fluid medium. So that... Oh, wow, look at that. It's got bacteria stuck to it or something. Look at this right here, right smack in the middle. Isn't that strange? See him spin? Probably Bacillus Bacteria of some species or another. Spinning around. Now that's cool. That's something you don't see every day. That thing is literally covered in those. Now I have to ask myself, what are they? Are those symbiotic bacteria attracted to it because it's producing perhaps oxygen in the fluid medium or something? Or giving off some kind of other nutrient? They don't look like flagella because they're not moving. That is really... Notice something else. This is a chain of bacteria here. You can see segmentation. It's hard to make out, but it's there. The longer one is comprised of about probably 10 segments, 9 or 10. Oh, that's beautiful. Look at that. Nice. Very arrow-straight Bacillus bacteria. Look at this. Spiral bacteria. Spirillum. Stuck in place, rotating aimlessly. That'll be a very common sight in the hours and days ahead as bacteria take over. Be more and more of those. There are already a lot of bacteria on this slide. See right there, slightly out of center. That's bacteria. There are already clusters of bacteria developing here and there. Let's clean that up a bit. More. Classic Diatom. Now here is an exceptionally good target. I was wandering around the perimeter of the petroleum jelly seal, which is always a great place to find things, and found this innocuous looking piece of junk, except it's not a piece of junk. It's a living organism. Piece of algae, large cell, larger anyway. Stepping up in the power, and back into focus, or out of focus rather. There it is, and already you can see all kinds of detail, and a discernible, a very obvious green color. Okay, now going for the gusto, the 40 power objective. Yeah. These are interesting. Some kind of algae cells. There's quite a lot of them. Diatoms. There it is. Okay, now there's some large cell algae. Now there is no mistaking the segments in the individual cellular structure. You can see quite a lot of detail inside the cells now. Very cool, huh? Now there are algae types that have much larger cells than this. This is pretty good. Exciting enough. See it moving? That's not me doing that. Once again, it's moving under its own volition. A lot of these types of creatures, they have mobility. You would be surprised. It's fairly common. Just because it's made up of a single cell doesn't mean it's a helpless victim of its environment.